Patient A has gone through extensive aerobic exercise without hydrating enough during the race. The patient has been dehydrated, represented by the 50% increase in concentrations of dissolved substances in the blood, as water in the body was released as sweat during the exercise. In contrast, glucose level increased by 50% without being decreased as part of aerobic exercise, meaning that the patient ate something during the race, while also breaking down glycogen using insulin. The process of ultrafiltration occurs between the glomerulus and the Bowman's capsule. The arterial blood contains water, ions, glucose, urea, blood cells, and other proteins.
The arterial blood is transported to the glomerulus via the afferent arteriole. The arterial blood flows through the glomerulus, a fenestrated capillary system.
Because the radius of the efferent arteriole is much smaller than the afferent arteriole, a pressure gradient is created.
Thus, at the glomerulus, a high hydrostatic pressure is created. This leads to water, urea, sodium, and glucose diffusing into the Bowman's capsule from the glomerulus. In essence, the net pressure gradient in the glomerulus supports the passive diffusion of the contents into the Bowman's capsule. Because proteins, including blood cells, are too big compared to the pores of the semipermeable membrane, they do not diffuse into the Bowman's capsule. Remaining substances in the glomerulus then flow to the efferent arteriole as postglomerular blood. The first process of reabsorption occurs in the proximal convoluted tubule, or PCT. The PCT contains filtrate from the Bowman's capsule and is next to tubule epithelial cells that aid transport of contents to the venous blood. The PCT has membrane-bound protein pumps specific to each filtrate. Sodium ions are actively transported into the epithelial cells via protein pumps. Glucose is either actively transported via protein pumps or co-transported with sodium via specific protein carriers. Because the concentration of filtrate is higher in the epithelial cells, this makes them hypertonic. Hence, water diffuses from the hypotonic PCT region to the hypertonic epithelial region via osmosis. The next process of reabsorption occurs in the loop of Henle. The loop of Henle dips into the renal medulla. The descending limb has no protein pumps for salts like sodium ions to be transported and only aquaporins for water transport. Salts and urea flow to the ascending limb. The ascending limb has no aquaporins for water transport, but only protein pumps, allowing only ion transport. Salts like sodium ions are actively transported out from the ascending limb of the loop of Henle for reabsorption in the venous blood. Because of the lower concentration of salts created by active transport, the loop lumen is hypotonic. Hence, water diffuses out to the interstitial fluid via osmosis through aquaporins in the descending limb. Urea stays in the loop of Henle at a high concentration to be transported to the next stage of reabsorption. The next process of reabsorption occurs in the distal convoluted tubule through reabsorption processes like that in the PCT. The final process of reabsorption occurs in the collecting duct. The filtrate solution now contains a concentrated solution of urea and sodium. The collecting duct is adjacent to tubule cells containing antidiuretic hormone, or ADH, receptors. Because this patient is dehydrated, much ADH is released from the posterior pituitary gland. By binding to ADH receptors, the production of aquaporins is increased. This essentially creates more channels for the diffusion of water out of the filtrate. As the level of ADH secretion is high, the resulting urine is more concentrated than that of a well-hydrated individual. The remaining filtrate flows down the collecting duct to be transported via the ureter. The collecting ducts are shared by other nephrons, which deposit concentrated urea solution in the collecting duct.
This concentrated solution flows down to the ureter to be secreted as urine.
Patient B receives hemodialysis. Because the patient does not have breakfast, the blood glucose level is lower. The blood urea, sodium ion, protein, and red blood cell levels are all at the standard level of concentration due to the patient's frequent dialysis. Hence, the dialysis fluid provided by the doctor will need to have the same ion and glucose concentrations as that in the blood. Because the urea concentration is higher in the bloodstream, urea is passively diffused into the dialysis fluid through the porous semipermeable membrane. Because proteins, including red blood cells, are too big compared to the pores of the semipermeable membrane, they do not diffuse into the dialysis fluid and remain in the bloodstream. Other ions diffuse in and out at the same rate, with zero net change in amounts due to the same concentration in the dialysis fluid and the bloodstream.